Imagine medicine that does not flood the whole body, but sends intelligent particles directly to damaged tissue. In this project, we explore a 2030 to 2050 vision. Nanobots detecting disease, clearing plaques, delivering therapy, and helping cells repair themselves. Across the previous projects, we built a chain of ideas: autonomous AI systems, spatial and brain-computer interfaces, bio-AI medicine, coordinated robotic swarms, and programmable materials. Now those threads converge inside the body. Project 23 asks how intelligent therapeutic particles could sense, navigate, deliver treatment, and support precision repair at cellular scale. The shift is from treating the body as one broad system to repairing specific biological problems where they begin. Nanobots or smart particles could sense signals, move through tissue, and release therapy only at the right site. They function like microscopic repair crews, clearing plaque, calming inflammation, helping tissue heal, or targeting tumors, guided by a personalized model of each patient. This venture is not just a particle, and not just an AI model. It is a closed therapeutic loop. Patient data reveals a disease signature, such as acidity, enzymes, inflammation, hypoxia, or specific proteins. AI translates that signature into a particle design, choosing the sensing chemistry, payload, dose logic, delivery route, and monitoring plan. Once deployed, particles act locally. They may carry drugs, RNA, immune modulators, regenerative factors, or contrast agents. Imaging and clinical feedback then update the next decision. That is the platform advantage. Therapy becomes measurable, programmable, and adaptable. Inside the body, delivery is never a straight path. Particles are carried by blood and lymph, then must leave vessels, move through dense tissue, and recognize the right biological signals. Some navigation is passive. Flow, leaky tumor vessels, permeability, and molecular attraction do part of the work. Other strategies add guidance through magnets, ultrasound, light, chemical gradients, or controlled external systems. Success depends heavily on particle design. Size, shape, surface chemistry, and charge affect circulation time, barrier crossing, and whether the immune system removes the particle before it reaches the target. Nanomedicine works best as a stack, not a single invention. At the foundation are materials: particles, polymers, lipids, gels, magnetic structures, or DNA-based shapes that can safely move through biology. Sensing adds value by detecting biomarkers, local chemistry, or treatment response. Intelligence then connects imaging, diagnostics, AI, and patient-specific models to decide what particle to use and when to intervene. Finally, control systems apply magnetic, acoustic, optical, thermal, or electromagnetic energy to guide, activate, or monitor the therapy. The power comes from integration. Therapeutic particles are a design space, not one product category. The core choices interact. Material determines stability and manufacturability. Size affects tissue penetration and immune recognition. Surface chemistry influences where the particle goes. Payload defines the clinical job, and triggers control when it acts. A liposome carrying RNA, a magnetic particle for localized heating, and a cell-mimetic vesicle for immune signaling are very different businesses with very different risk profiles. The founder's question is not which nanoparticle is best. It is which combination delivers the needed function, then safely disappears, degrades, deactivates, or can be retrieved. Biomarker sensing turns a particle into a context-aware decision maker. As it moves through tissue, it reads local cues such as pH, oxygen level, inflammatory cytokines, enzymes, and receptors. Healthy tissue may show one harmless signal, while disease sites often show a combined pattern. That is why many designs use AND logic, activate only when several conditions match at the same time. Patient data from molecular diagnostics or liquid biopsy can guide which signals to watch. The result is more selective release, fewer off-target effects, and the possibility of confirming that activation occurred. Targeted delivery changes the goal from flooding the whole body to controlling where, when, and how a therapy acts. Passive targeting uses biology, such as leaky tumor vessels or natural tissue accumulation. Active targeting adds recognition through ligands that bind specific receptors on diseased cells or inflammatory sites. Triggered release then provides timing control, so particles unload only when they sense heat, light, ultrasound, enzymes, pH, magnetic fields, or local biochemical cues. The next layer is AI-guided dosing, which uses imaging and response data to adjust treatment for each patient. At this stage, AI becomes the coordinator of the therapeutic loop. Imaging methods such as MRI, ultrasound, PET, endoscopy, and digital pathology reveal where disease is located and how it is changing. Molecular diagnostics add blood, tissue, genomic, proteomic, and metabolic signals, showing what the disease is doing biologically. AI combines these inputs to choose particle type, targeting chemistry, payload, dose, and release timing. After treatment begins, monitoring data are compared with the expected response. If repair is too slow, inflammation persists, or biomarkers shift, the system updates the next intervention. A health twin turns particle therapy from a generic intervention into a patient-specific design problem. It combines anatomy, disease state, molecular markers, immune status, and treatment history into a living model of one body. Before particles are used, clinicians can simulate where they travel, how likely they are to hit the target, and where toxicity might appear. The prescription can then adapt to age, genetics, microbiome, organ function, or disease subtype. As monitoring data returns, the twin updates, so each adjustment is better informed than the last. Repair can be viewed as a spectrum. First, detect the problem, then intervene locally. In cardiovascular disease, particles might image plaques, calm inflammation, or target clots. In cancer, they could read tumor signals and release drugs only where needed. In the brain, the challenge is crossing barriers and addressing inflammation or aggregates. In regeneration, particles become instructors, delivering growth cues, genetic regulators, or scaffold materials that help damaged tissue rebuild itself. External control turns the particle platform into a responsive treatment system. Magnetic fields can pull or concentrate magnetic particles near the target. Ultrasound can help particles move deeper into tissue, trigger release, or create local mechanical effects. Light, heat, or electromagnetic energy can switch payload activity on only where needed. The key is closed-loop control. Imaging confirms where particles are, software adjusts the field or trigger, and safety monitoring helps prevent activation in healthy tissue. This is how precision improves without adding another invasive step. By 2050, the biggest shift may be from treating disease after it disrupts the body to maintaining health continuously at the microscopic level. Early nanosensors could flag molecular warning signs before symptoms appear. Targeted particles could then deliver drugs, repair tissue, or trigger regeneration only where conditions match the patient's biology. Hospitals may become design and coordination centers using imaging, digital health twins, and biofabrication to plan interventions. This future also raises hard questions. Who controls autonomous systems inside the body? How consent works over time? And how we prove long-term safety? The core takeaway is that repair medicine becomes powerful when particles, sensors, diagnostics, AI, external control, and health twins work as one system. That shifts care from broad treatment to precise, responsive intervention. The same logic now scales outward. Next, we apply programmable materials thinking to energy fabric, then to autonomous climate repair and carbon mineralization networks.